Okay, this lecture is about the mechanism of DNA replication. It's probably going to be a little on the long side, so always feel free to pause whenever you need a break. But we've got like 3 slides worth of outcomes to cover, and you will see we'll go through the outcomes at the end. So, one of the challenges of learning DNA replication is that we draw it and we draw it and we draw it, but it's really a dynamic process, which means it's in motion. So I highly suggest that you watch these videos here in the green box. Animations can be very useful for helping you visualize what's happening. The first one is a nice step-by-step video. If you have questions about some of the terms they use, it's a little bit older, you can always ask, but you should be able to get the general gist of the proteins that we are specifically talking about, and some of the ones that we're not talking about. The Amoeba Sisters, this is kind of a cute, fun video on DNA replication. And this other one, woo, at the bottom is a really good basic. It has some extra information again, so just always focus on the information in our outcomes. So again, the outcomes are listed at the end. Here are some really big important concepts that you need to understand, and we will talk about these in the upcoming slides. So some key terms. So first key term is DNA replication is called semi-conservative. Okay? And semi-conservative means that each new DNA double helix is made of one parent strand, shown in red, and, let's see if I can switch colors, one daughter strand, shown in blue. Okay, so it's kind of semi-conservative in that we've conserved one strand and we've made one new strand. We've conserved one strand, we've made one new strand. So up here is what we call the parent strands. This is the original double helix. And then we're going to talk about how we split the helix open. We break the hydrogen bonds. We open it up and we copy, or DNA polymerase copies, and makes a new daughter strand that is complementary by base pairing to the parent strand. So now these new double helixes are identical, right? CG, CG, AT, AT, CG, CG, TA, TA. So we have made a exact copy and by using the parent strand as a template, or that's how we make the exact copy, I should say, is by using the parent strand as a template. And this was discovered by 2 scientists, Nelson and Stahl. So be sure to watch the scientist or DNA discovery scientists and experiments video. We're not going to have time to go through that in class, but you are responsible for that information on quizzes and exams. Okay, so DNA replication is semi-conservative, kind of conservative. And I just want to let you see that if you took these 2 strands, right, that we had here, and now replicated them again, you would split them. Okay, so the red goes to the red, And the blue is at the blue. And then let's use purple to make the new daughter strands. Right? So these new double helixes would still be semi-conservative. They have one parent strand and one new daughter strand, and one parent strand and a daughter strand. So you continue and do this every time the cell divides. It's all based on base pairing, okay, or complementation or complementary. So during DNA replication, the cell is making a complementary strand, okay, due to the base pairing rules. And remember, the strands are always anti-parallel. So I want you to pause the video, take a second, and write the complementary strand to this strand of DNA. And the complementary strand, we'd start at the 5' because we're going to learn DNA polymerase only works 5' to 3'. And as we make the base pair, we say A, base pairs with T, and G, and T and A, and C and G, and A and T and A, and G and C, and A and T, and this makes the 3' end. So they're anti-parallel, they're running opposite ways, they're all made by base pairing, and if you wanted to put in your representation of hydrogen bonds, you remember that the AT base pairs make 2, and the GCs make 3. Okay, so be able to do this, on top of this, be able to do it recognizing the DNA nucleotide structures. So that's in the structure lecture. If I gave you this set of nucleotides, this nucleic acid strand with their molecular structure, you should be able to figure out the complementary base pairing and know that they're running anti-parallel. Okay. So let's talk about how DNA replication happens. So first of all, here's a nice double helix. We don't draw it all twisted because that just gets too complicated. You notice it's anti-parallel, 5' to 3', 5' to 3'. And so somewhere here is going to be what we call the ORI, or the origin of replication. So it's where a protein called helicase, which we'll talk about in a minute, opens up. So this is the starting point for DNA replication. And it's kind of interesting, you might be asking, why does it happen in the middle? And what I will show you eventually is that our long chromosomes have multiple origins. So they have multiple start sites for DNA replication, because if you tried to just go from one end to the other, it would take too long. Excuse me. Also, I want you to note that origins of replication have high AT base pair content. So if you looked at the nucleotides here, there would be a lot of AT base pairing. And that's because A and T only have 2 hydrogen bonds between them versus G and C, which have 3. So it's a little easier to break through these hydrogen bonds, to break them apart when there's only 2 per base pair than 3. This thing is called a replication bubble. Each of these parts are called replication forks. So as we draw DNA replication, we're just going to look at one fork, one half of a replication bubble, but understand that there are two replication forks, and DNA replication goes in two directions. So we'll talk about that in a minute, bidirectional replication. So how does this happen? So the protein that binds to the origin that helps start unwinding is called helicase. So let's look at this next slide. All right, so we have this blue protein called helicase. It's an enzyme because you see the ASE ending, and its job is to break hydrogen bonds basically splitting the base pairs and opening the double helix. Now, the double helix is very stable in this base paired structure. So what has to come on is proteins called, we call them SSB, single-stranded binding proteins. And they're going to coat or bind to the open DNA, the single strands, so that they can't base pair back together. Right? It's kind of like opening up Velcro, and if you're not careful, the two pieces will go right back together and stick again. So you've got to block the Velcro from interacting. So you're blocking all the nucleotides from being able to base pair with the other strand. So helicase is opening up the double helix so that we can come through and copy these strands. There is one more very important enzyme called topoisomerase. This is the blue one, and I want you to notice that it's in front of the helicase. And it's hard to illustrate, so I really suggest you watch this video on topoisomerase. The video shows you the idea that if you took two pieces of a rope and started splitting them apart, right? So you have a rope that's twisted around each other, you split it apart. As you're splitting it apart, the part that's still wound up is getting tighter and tighter and tighter, right? Because it has this strain of you opening up that twist. And so what topoisomerase is, as it says right here, it alleviates the coiling caused by opening up or unwinding the helix. And it's pretty amazing protein. It will actually cut your DNA and kind of, like if you were letting string unravel a little bit, and then put it back together. Okay. So it has to relieve that strain by actually breaking your DNA. One other thing I need to note for you, and that is that we are going to talk about eukaryotic proteins. So if some of the terms I use are a little bit different than your textbook, for instance, in bacteria, topoisomerase is called gyrase. Okay. I'm teaching you the eukaryotic proteins. If you take microbiology or genetics, you'll probably learn both. Well, microbiology, obviously you'll learn prokaryotic proteins, and genetics, many times we teach both prokaryotic and eukaryotic. They do the same functions, they just have different names. Okay, so we've got helicase, we've got single-stranded binding protein, and topoisomerase. once we have the DNA unwound and a single strand of DNA is being exposed, we can now copy it. Okay. So the enzyme that does the copying is called DNA polymerase. So this is an enzyme that makes DNA. This is the enzyme that makes the phosphodiester bonds between DNA nucleotides. Okay. And it's going to copy by base pairing, and it moves along this single strand of DNA, and there's nucleotides floating around in the nucleus, and they will come in, and if they fit by base pairing, DNA polymerase will hook them together, and then the next one, it will read the next nucleotide and the next nucleotide. DNA polymerase can only attach nucleotides to a 3' hydroxyl group. Look like one word there. Okay. It needs that 3', and remember we draw our nucleotides, sugar, phosphate, base. Okay. It requires this 3' hydroxyl group, and it only attaches to the 3'. So we call the direction of DNA replication 5' to 3'. So this is describing the new daughter strand. It is made 5' to 3'. So you're always adding to the 3' end. It's going to copy the parent strand shown here in red from the 3' end to the 5' end because they're anti-parallel. So the new strand is being synthesized. Oops. Oh, why does it do that? The new strand is being synthesized 5' to 3'. Okay? So this is always talking about the new daughter strand. So where does DNA polymerase get a 3' OH? How does it start? And DNA polymerase cannot actually start by itself. It has to have what we call a primer. Okay? So DNA polymerase, this should say, cannot start synthesis without a 3' OH. So we need something to help it. We need a primer. And the primer is actually made out of RNA. So RNA is also made out of nucleotides. Phosphodiester bonds can base pair, and RNA polymerase let's say polymerases, any kind of RNA polymerase can start what we call de novo. It can start without any nucleotide. So it can start from scratch. And the special type of RNA polymerase that makes the RNA primer is called primase. We're just going to leave it at primase. Some books call it RNA primase. Some books call it DNA primase. We're going to just call it primase is the enzyme that makes the RNA primer. Oops, I can't put that on there. Too low on the screen. Makes RNA primer. How about that? So in fact, there would be a protein here called primase that would be synthesizing this RNA primer. And again, it goes 5' to 3'. Every synthesis of RNA or DNA is 5' to 3'. Once RNA primase, put a darker color here, makes this RNA primer, there's a 3' OH, because even RNA nucleotides have that 3' OH. Okay. So now there's a 3' OH, and now DNA polymerase can come in and start synthesizing DNA. Everything happens 5' to 3'. I want to back up one slide because I forgot to tell you. The DNA polymerase that makes DNA for the majority of replication is called DNA, let me write this over here, DNA Pol, short for polymerase, this little thing, which is a delta. Okay, that's the eukaryotic name for the DNA polymerase. So if you see this little squiggly guy, delta, that is the main replication enzyme. Okay. So we now know that you have to have an RNA primer before DNA polymerase can start. And we're going to add some more terms. And we're going to add the terms leading and lagging strand. These terms describe the daughter strand. I want you to notice they're both made 5' to 3', 5' to 3'. Okay. The leading strand is made towards the replication fork. The lagging strand is made away from the replication fork. Okay. Both start with an RNA primer. Both are using DNA pol delta as the polymerase. I'll show you why we call the lagging strand lagging in a second, but I want you to be able to recognize leading versus lagging strands. So we've got DNA replication happening on both strands at the same time, but because DNA polymerase can only go one direction, right, the strands are made in the opposite direction. So if we opened up, so here's our slide from before, our image from before. If we open up that replication fork a little bit further, right, so we've helicase is working, it's unwinding. Here's helicase. the leading strand can keep on copying, right? 5' to 3', it just keeps going and going. But the lagging strand must start over. That means that as the replication fork opens, we have to make a new RNA primer, and then we have to put DNA polymerase back on to copy some more DNA. So do you see how this is continuous? And down at the bottom here, this is discontinuous. So we call DNA replication semi-discontinuous. Someone asked me one time why it's not called semi-continuous. I have no idea, but that would make sense as well. But it's called semi-discontinuous. So it's kind of discontinuous. It's continuous and it's discontinuous. So altogether it's semi-discontinuous. Again, everything's anti-parallel and it's all based on base pairing. So if we opened up the DNA replication fork even more, we would continue making the leading strand, and we would continue restarting the lagging strand. And all of these small strands are called Okazaki fragments. So every time you have to restart DNA replication for the lagging strand, you make a new Okazaki fragment that eventually is joined together to make a complete lagging strand. So it shows here, if we look back really quick, we had two Okazaki fragments, fragment 1, fragment 2, and then, these two got joined together, and that's what we're going to talk about next. So eventually, RNA primers are removed and the Okazaki fragments are joined. And we'll talk about how that happens in just a second. Okay. Again, every time you make a new piece of DNA, you have an RNA primer shown in yellow. So, semiconservative is the idea that a new double helix is a parental strand plus daughter strand. It conserves the parental. The daughter is new, so it's semi-conservative. Semi-discontinuous talks about the idea that there is a leading and a lagging strand. Anti-parallel means that the two strands go in opposite directions. 5' to 3', 3' to 5'. It's all made by complementary base pairing. A base pairs with T, T base pairs with A, G base pairs with C, C base pairs with G. So that's how we can accurately copy one strand of DNA because of base pairing. Okay, next term, bidirectional. So we talked about An origin of replication is where the helicase starts. Okay? So helicase is now here unwinding, right? But it started up here. And as soon as it started unwinding, we had an RNA primer come in, and we had DNA replication happening. And I know it goes this way because I know it goes 5' to 3', and this strand is running opposite 5' to 3', 5' to 3'. This strand is running opposite 5' to 3'. I'm just showing the leading strands right now to illustrate this idea of bi-2-directional. So DNA replication happens in both directions. So you have a replication bubble that initiates at the origin, and it results in 2 replication forks. So everything we just showed you before is happening in both directions. So this is a nice little image to illustrate what's going on. So remember, wherever you have a leading strand, you have on the opposite side a lagging strand, and a leading strand and a lagging strand. So here we normally split it and we show 1 replication fork, but it's really happening twice. You actually have 4 DNA polymerases working. Right? You got 1 here, you got 1 here, you got 1 here, you got 1 here, and every time you open this fork up, you start with another one. So I guess I shouldn't show them here. I guess they were really the last time they were here. Let's be a little more accurate. Okay, 4 DNA polymerases working at once. You have 2 helicases opening up. And you also have 2 topoisomerases helping undo that strain by opening up the helix. So lots of stuff is happening, and this is all because DNA polymerase can only go in one direction. And because origins of replication are in, I'm going to say the middle, but it's not the exact middle of a DNA strand. I highly suggest you practice drawing this. You practice recognizing your 5' and 3' ends of all of your strands, both parental and daughter. So you might be kind of wondering how this could all really happen. And DNA replication is obviously a little more complicated than what we are drawing. And it really happens by something called the trombone method, which is kind of illustrated here, that the leading strand up top here is continuously produced. Okay, so here is your DNA polymerase. You're going to see something called a clamp that just holds the DNA polymerase on. You've got 2 DNA polymerases working on 1 replication fork. Here's your helicase, right? And we'll talk about these guys down here in a minute. I highly suggest you watch this. This is a really good video of DNA replication. Really good. Not a really god. A really good video. And this one is a really nice illustration or animation showing the trombone method. So take an extra few minutes and watch those videos. Okay, so we talked about semi-discontinuous replication. Now we're going to talk about what to do with those RNA primers. Because your DNA and your chromosomes are actually fully DNA. They don't have RNA in them. So what do we do with those RNA primers? They are removed. So we have two important enzymes to talk about next. We have DNA polymerase alpha, and we have an enzyme called ligase. Okay. So let's look down at this bottom strand here. You had a lagging strand, and between each lagging strand is your RNA primer. So this is showing DNA pol delta hooking onto the RNA primer and making more DNA. Once it bumps into this RNA primer, it stops. But then we eventually need to get rid of this RNA primer because we want it to be continuous DNA. And so we have a second DNA enzyme called DNA pol alpha that comes in and basically chews up the primer and replaces it with DNA. This is again where you really need to watch an animation. Okay. DNA ligase, and I'm going to write this all in the next slide, is the enzyme that makes the final phosphodiester bond. So it hooks these two different DNA strands together and makes the final connection. So let's zoom in and look at this. So DNA polymerase alpha removes RNA primers and synthesizes or makes the DNA that's missing. So it's replacing the RNA primer with DNA. But it can't make the final connection. So DNA ligase, we say, seals the nick. Nick is like a cut. So it makes the final phosphodiester bond between two DNA strands. Okay. So, got a lot of enzymes. You need to memorize their functions and know where they work. Be able to explain their functions, be able to know what would happen if they weren't working properly, and be able to identify them on different diagrams. Okay, so I never give you the same diagrams I give you for class notes or online lectures because that's pure memorization. If I give you a different looking diagram, then you're actually applying your knowledge. So here's a couple questions to quiz yourself. Name the protein pointed to the brown circle. So what you should be looking at is, okay, this is the leading strand, 5' to 3 prime. This must be my DNA polymerase. And if you wanted to be super accurate, it would be DNA pol delta. Topoisomerase would be up here somewhere, right? It's in front of the replication fork. Helicase would be right here at the replication fork, unwinding that DNA. And primase would be making these RNA primers. So whenever you see a different color on the DNA, that's representing an RNA primer. Okay, so look at this diagram and name the protein pointed to the brown circle that's making the red strands. That enzyme that's making the red strands, it's making the RNA, is called primase. Okay? So even if these diagrams look a little weird to you because they're already showing DNA replication happening, just understand, just think about, this is the RNA primer, this is the enzyme that's making it, that's called primase. Which image shows semiconservative DNA replication? The answer is B. So what you need to look at is parent now paired up with a daughter, blue, a new color. Parent are still now paired up with daughters, they're just showing the daughter as always being blue. Okay. This one is called conservative because the parents always stick back together. See how the parent went back to the parent and this parent went back to this parent? And this one is called dispersive, which means each strand is a little bit of a mix of parent and daughter. And we know that these are not the way that DNA replication happens. And again, this was discovered by Messelson and Stahl, so make sure you watch the video about their research. Okay, which letter is which represents the leading strands or strand? So the way you want to tackle this is the one that points to the replication fork and that is continuous are going to be the leading strands. So that's A and H, so the answer is D. Okay, which are the 5' ends of the parent DNA? So if these are the daughter strands being made, and you know they're being made 5' to 3', then you know the parents have to go in the opposite direction, 5' to 3'. And this one's being made 5' to 3', so the parent is 5' to 3'. The parents are in the darker blue. So A and D are the 5' ends of the parent strand. The answer is C. All right, we just have a couple more topics, and then we are done. So I want you to understand that DNA replication is very accurate. We're not going to go into this, but I can't teach DNA replication without you understanding that it's very accurate, but mistakes are made. So you can barely see this guy here, this little Pac-Man. That's your DNA polymerase. And every once in a while, the wrong nucleotide will be incorporated into the daughter strand. And what happens is that the DNA polymerase can sense this. So it's called a mismatch. So say the parent strand was G and there was an A put in there. Well, they're not going to be able to base pair. Or I should say incorrect hydrogen bonds. That's what holds them together. So DNA polymerase can actually move backwards. It can backtrack. So it's DNA polymerase, delta, will backtrack and cut out, so break the phosphodiester bond, do hydrolysis, mismatched nucleotide, and then continue on. Okay. So we know that replication goes 5' to 3'. So this backtracking, removed by proofreading, is something called a 3' to 5' exonuclease activity. Exo means at the end. There are enzymes that are called endonucleases, E-N-D-O. That means they cut inside. Nuclease means it is removing, or it's breaking phosphodiester bonds. And so this 3' to 5' exonuclease activity allows the DNA to correct mismatches. It allows what we call proofreading, kind of like backspace when you're typing and you make an error. Therefore, normally the error rate is approximately 1 error in every 10 to the 7th to 10 to the 8th base pairs. Okay, so 10 to the 7th, 2, 3, 6, 7, 2, 3. So every 10 to 100 million base pairs, there'll be a mistake that actually isn't fixed. If you didn't have proofreading, this error rate would be much, much higher. Something I want you to think about is that your genome has about 3 billion nucleotides, 3 billion base pairs, I should say. So every time your DNA replicates, even with this proofreading, you can still accumulate at least 5 mistakes. Okay. We call these mutations, and they may or may not have an effect on protein expression, which we'll talk about in transcription and translation. They may or may not have an effect on gene expression, we'll talk about for transcription. So every time your cell divides, it accumulates some errors. And these errors are not always fixed, and that means they're passed down. And eventually, if you accumulate errors in the right genes, or maybe I should say the wrong genes, that's what produces cancer. Cancer is really a disease of cell replication due to DNA mutations. Now, not all cancer, but as you get older, you accumulate mutations, and that's why as you get older, you have a higher risk for cancer. It's just kind of natural part of biology. I think it's pretty amazing that it can be as accurate as it is. Okay, next topic, I just want to show you the difference between eukaryotic and prokaryotic DNA replication, and this leads us into the another lovely topic of why you will eventually die. Okay, so eukaryotes have linear chromosomes. That means our chromosomes are in a line. Prokaryotes and your mitochondrial DNA, and if you were a plant, your chloroplast, are circular. And the reason this is interesting is because prokaryotes, your mitochondrial and your chloroplast DNA, only have one origin of replication because their genome is much, much smaller than ours. So you can open up, and in fact, DNA replication happens bidirectionally, but it copies one strand going one way and one strand going the other way. So there are no lagging strands, just leading. Because they can eventually meet, right, and come together, and now you have two replicated DNAs. So their replication is continuous. They still need an original RNA primer, but what happens is when this one comes up, it just removes the primer and connects into a full circle. Okay, pretty cool. Versus your DNA is so big, even in the 46 chromosomes you have, that we have multiple origins of replication. That's why we have these replication bubbles and we have bidirectional. But then we have something called an end replication problem. I want to show you that. So, if you look here, this side of the figure first. Here's our replication bubble. We're copying. We eventually remove all of those primers. The primers are red. Okay, and when we remove these last final primers, what I want you to see is there's a gap. We can't copy this last little bit of DNA because there's no 3' hydroxyl hanging out there. And so we have a gap. And that means every time your DNA replicates, it gets shorter. So every time DNA replicates, we lose a little on the daughter strand. You're losing DNA every time your cells replicate. Okay. The chromosomes get shorter. And this is a nice zoom in, kind of showing you the issue that once we remove these RNA primers, right, we can't fill in this gap. We call this the end replication issue. So if this is making you a little concerned that your cells are losing some DNA information, it should be. Okay? This is a concern. And what we have, oops, there's the highlight to look at. What we have evolved with on our ends of our DNA is something called a telomere. So telomeres are sequences of repetitive DNA at the end of eukaryotic chromosomes. They're repetitive sequences. You can kind of see GGG-TTA. This is a short one. They're really actually 100 or so nucleotides long. Okay. And they're adding on to the end. The point is, the purpose is to deal with the continuous loss of DNA due to replication. So let's look at this a little bit more. Okay. So you have an enzyme called telomerase. So this is the enzyme that makes telomeres. And what this enzyme does is it uses an RNA template, and it adds on some DNA to the ends of the chromosome. So you can kind of see this. Telomerase synthesizes this nucleotide repeat region, okay, and it adds hundreds of nucleotides to the end of the DNA chromosome. So it's making a repetitive sequence here. This RNA primer still has to be lost, so we still have an end with a gap. But what we do in our cell is we fold that end around and make a little cap to protect the DNA. Okay, so the ends of telomeres are folded back on each other to protect it so it doesn't get destroyed by other nucleases. Telomerase, this enzyme, is active in stem cells, and stem cells are kind of like the baby cells that you have that generate all of your new cells. It's active in egg and sperm producing cells. But it's not active in adult cells. So once your cells become, say, a skin cell or a liver cell or a muscle cell, the telomerase is shut off. And that's when your telomere shortening starts to happen. There has been a lot of research about, well, hey, could we turn on telomerase? Because what happens is when your chromosomes get short to a certain point, it signals the cell that we're going to start losing important genetic information. And it signals the cell to go through apoptosis. And apoptosis is called programmed cell death. That's why you're going to die eventually, even if you're totally healthy, because eventually your telomeres are going to get so short that the cells self-destruct. And some organ of your body will fail. When you turn on telomerase in normal cells, you get cancer. So telomerase is active in many types of cancer. That's why cancer can go on and divide forever and ever and ever. So you do not want to turn on your telomerase in all of your cells. It's a fact that as you get older, years, your telomere length gets shorter. Now, there are some companies out there that say, oh, give us a sample of your cells and we'll tell you your biological age based on your telomeres. So say here I am at 50, right? Say they do my telomeres and say, oh my gosh, your telomeres are really short, you've lived a hard life, you're really of the biological age of 75. Or maybe I get good news, oh, your telomeres are really long, you're biologically only 20. Well, first of all, there's nothing you can do about it. Second of all, when you have shorter telomeres, there are companies that try to sell you products that will, they say, build back your telomeres, increase the length, and turn on telomerase. You don't want that. First of all, it doesn't work. And second of all, if you turn on telomerase, all the science points to cancer. So, I don't care how biologically old I am. I'm going to live my life day by day, right? And when it's my time, it's my time. Okay, based on that happy news, let's talk about one last topic. And I just in general want to remind you about DNA structure, the difference or the relationship between the DNA double helix that we're drawing and chromosomes that we're going to talk about next unit in genetics. Okay. And they're really the same thing. It's just that your DNA is organized, your double helix is not floating around because it would get tangled and broken. It's organized by proteins called histones. That's what these little orange guys are. And the organization of DNA around a histone is called a nucleosome. And I highly, highly suggest you watch this video. It's really good showing this organization and how our double helix gets compacted, wrapped around these histones, and then they get compacted and they get compacted, and we get our chromatin, which is DNA and proteins. And eventually, that chromatin can pack nice and tight into a chromosome, and our cells can go on and divide. Okay. The reason I bring this up is because there are two final terms on your objectives, and that is euchromatin and heterochromatin. Heterochromatin is tightly wound DNA. Lots of histone proteins, no room for other proteins to get in and read that DNA. And the next topic we talk about is transcription or gene expression. Euchromatin is open for gene expression, for transcription. And we symbolize transcription by a bent arrow, and you can see down here, this with a bent arrow with an X means no transcription, no gene expression. And so the reason this concept, how do I get out of here? The reason this concept is so important is something I want you to start thinking about. This is one way that allows cells with the same genetic information. So all your cells in your body pretty much have the same genetic information to have different functions, to be of different cell types. Okay. Because different cells are going to have different genes in heterochromatin and euchromatin, which means different cells have different genes being expressed and not expressed. So keep this concept in the back of your mind. It's really important for this unit. As we get into transcription and translation, we'll talk about the proteome versus the genome, but this is really an important part of that. Okay, let's go through the outcomes real quick and make sure we covered everything. So, Be able to generate a complementary sequence if given a template. So if I give you 5' ATGC, you need to give me 5' to 3' information. Okay. So explain how semi-conservative replication works. We've talked about that. Know the experiments. That's in a different online lecture. We've talked about bidirectional and what this means for the different daughter strands that are synthesized. So remember, take a look at this image where you have both leading and lagging. Can guarantee there'll be questions like that. Describe how the replication fork opens, moves, and stays open. So it opens at an ORI by helicase, and it stays open because of those single-stranded binding proteins. And the replication fork can move because of topoisomerase. Describe how a daughter strand is synthesized. We've talked about that. We've talked about leading and lagging strands. Explain how the structure of DNA suggests a model for its replication. So this is trying to get you to pull all these concepts together. Anti-parallel and base pairing. Semi-conservative, we've talked about. Oh, we got that twice. Must be important. This is a nice slide you can print out to help you draw. Right? I've got 5' and 3' things labeled. You go through and try to draw the daughter strands, the leading, the lagging, where all the enzymes go. Explain why DNA ligase, we've talked about that, is putting together those Okazaki fragments, actually any DNA fragment, so at the very end, leading and lagging strands will be connected by ligase. We've talked about single-stranded binding protein, primase, and making the primers. Oh, that didn't work. DNA, delta, and alpha. Leading, lagging strands, Okazaki fragments, forks and bubbles, and helicase and topo. Okay, so this is where I suggest use these terms on this figure above. Try it out. Semi-discontinuous, we talked about because of leading and lagging. Polarities are talking about 5' and 3'. Nope, I'm going to put another picture. Understand the end replication issue. So this is the loss of DNA and, whoops, oh, I hate it when I do that. And well, that's the end replication issue, the loss of DNA. Explain the role of telomeres, which are your repetitive ends, and telomerase is the enzyme that makes it. Know the difference between euchromatin and heterochromatin, and describe the structure of double-stranded DNA from nucleotide to chromosome. So the main terms I want you to know here are histones and nucleosomes. Okay? And that they help organize the double helix. Alright, I'm scared to see how long this video is, but again, hopefully you paused it, took notes, and are working on DNA replication. Alright, thanks.